Again, just a quick recap from the previous video. So, type 1 precursor is found in the secretions such as saliva, body fluids. For the type 2 precursor, it is found in the red blood cells and is responsible for ABO antigen formation. Then the glycosidic linkage determines whether a precursor functions in body fluids or on RBCs, and then both precursors must be modified by enzymes such as your glycosyltransferases to express the ABO antigens. So in blood banking, precursor substances are the foundational building blocks used to construct blood group antigens. These substances consist of oligosaccharide chains, which are short strings of sugar molecules that are attached to either a protein or a lipid carrier molecule on the red cell membrane or in body fluids. The classification of these precursors into type 1 and type 2 is determined entirely by the specific chemical linkage between the two terminal end sugars, such as the D-galactose or the GAL, and then the N-acetylglucosamine or the GLC and AC. Type 1 precursor substance, the number 1 carbon of the terminal GAL is linked to the number 3 carbon of the GLC and AC. This is known as the beta 1,3 linkage. So these structures are found primarily in body fluids and secretions such as saliva and milk, and in the plasma. So type 1 chains are the mandatory precursor for the Lewis blood group system. The Lewis gene, or the FUT3, adds a sugar L-fucose to the number 4 carbon of the GLC NAC on this specific chain. So now let's proceed to the type 2 precursor substance. So the number 1 carbon of the terminal GAL is linked to the number 4 carbon of the GLC and AC. This is known as the beta 1,4 linkage. So this is the predominant type found on red blood cell membranes, while some type 2 structures exist in secretions. They are the main backbone for antigens actually attached to the red cell. So beyond serving as the base for A, B, and H antigens on the red cell, type 2 chains, specifically a structure called paragloboside, function as the mandatory precursor for the P1 antigen. Now let's talk about the formation of your H antigen. This is the only antigen in the H blood group system. So if an individual is a blood group O, it inherits at least one H gene, the genotype homozygous big H or the heterozygous big H or small h. So the H antigen is the precursor structure on which the A and B antigens are made. So inheritance of the H gene results in the formation of your H antigens. So both the big H and the big S small e genes are located at chromosome 19, but they are not really a part of the ABO system. So their inheritance influences the A and B antigen expression. So let's talk about it further. So the formation of the H antigen is a critical biochemical process because it serves as the essential building block or precursor upon which the A and B blood group antigens are constructed. So the production of the H antigen is controlled by the H gene, also known as your FUT1, which is located on chromosome 19. So most people, over 99.99%, inherit at least one functional big H gene. So genotype homozygous big H or heterozygous big H small h. So the H gene does not produce the H antigen directly. Instead, it codes for the production of a specific protein called an enzyme, which is the alpha-2-L-fucosyltransferase. This enzyme acts as a biological builder that carries out a specific chemical reaction. So to create the H antigen, the enzyme performs the following steps. So first, it identifies a base structure on the red cell membrane known as the precursor substance, specifically type 2 oligosaccharide chains or paragloboside. So the enzyme facilitates the transfer of a simple sugar called L-fucose to the terminal galactose of that precursor chain. Once the L-fucose is attached, the structure is officially recognized now as the H antigen. So why the L-fucose matters? Your L-fucose is known as the immunodominant sugar for the H antigen. This means it is the specific molecule responsible for H specificity. Without this sugar, the structure cannot function as a foundation for the ABO system.